All right, so let's get started, or let's continue. Okay, so in the final version of the lecture notes, you'll see the proper MATLAB plot of this thing. But if any of you want to see the raw data before that, I'm happy to share it with you. Just ask me. So everything we've been talking about here is sort of in terms of active systems, because we've been looking at things as a function of the system size. And if we're interested in materials, we really want to look at the quantities on a per volume or per mass basis. So what we're going to do is we're going to define a metabolic density. And this one is going to be little q dot. So the whole system was capital Q dot. That's the total heat rejected by the system. And it's the density of heat production per volume. The density, well, I don't need to say it per words. I'll just say it is Q dot over V, the system volume, okay? And, the reason I'm doing this is I have a hypothesis. This is sort of my active materials hypothesis, is that this quantity matters, okay? Right, my hypothesis is that Q dot is, or maybe might be is a better way. How about I say might be and more careful? No, it's a hypothesis, so I can say is, because I said hypothesis first. Is an essential parameter for characterizing active materials. Okay, so and I chose to do it per volume instead of per mass just because it's easier to compare with some calculations that I'll be doing later on, and to make the conversion from mass to volume, I just assume that all the organisms have the same density and that it's basically the density of water, and with this dynamic range, doesn't matter, okay? So I'll plot up Q dot in less detail than I did little q dot in less detail than I did big Q dot. So little q dot has units of watts per meter cubed. And I'm going to plot it against a length scale, which is the volume to the 1/3 power with units of meters. And the smallest length scales here are 10 to the minus 8, about 100 nanometers, up to something a little bit more than a meter. So we have about 9 decades on the horizontal axis. And on the vertical axis, we only have about 3 decades. It goes from 10 to the 3 to 10 to the 6th, okay? So roughly at 10 to the minus 8, it hits about 10 to the 6 watts per meter cubed, and down at around 1 meter cubed, it's about 10 to the 3 watts per meter cubed. And this has a slope that I measured, in MATLAB to be approximately minus 1/3, not 1/3, minus 1/3. Okay, so it's a relatively weak dependence on the radius of the object. So so the key thing to remember here is, is that a big dynamic range or a small dynamic range? I don't know. I would argue that it's surprisingly I would say it's a surprisingly small range of metabolic values. Like if you look at Young's modulus, you can measure Young's moduli as small as 1 Pascal and as big as 10 to the 12 Pascal. So, 12 decades. Here, if we look across all the living systems on average over, 25 orders of magnitude in mass, it only varies by 3 orders of magnitude. So I say that's a pretty small range. That's my opinion. So, but it varies over about 3 decades from one, and these are numbers, I'm writing them out. I'm going to use them all over and over again later on in our other calculations of 1 kilowatt per meter cubed, which roughly corresponds to a human, and 1 megawatt per meter cubed, which is a bacterium. Yeah? Are they what? Yes, they are. So because let's see, we can do the math. Okay, so Q, big Q dot is equal to little q dot times V. Okay, and so Q dot is equal, and I saw empirically that big Q dot was like V to the 0.9. That was big Q was V to the 0.9. Sorry, equals, or Q dot V is like that to the 0.9, so Q dot should be like V to the minus 0.1, but if that's R cubed, and I do minus 0.1 of R cubed, I get R to the minus 0.3. So they're compatible. Does that make sense? And I measured it before I calculated it, so I really trust it. But I only, actually no, but I only trust it when I can do both. That's right. But yeah, it works out. I was actually, it really bothered me that it didn't make sense at first, and I went through it. It's fine. Okay, so fun facts. Okay, these are the fun facts. I love these fun facts. I'm very proud of them. Okay, this one's fun. You know, if we had the same Q dot as bacteria, we would need to eat about 500 kilograms of sugar per day. That's a fun fact, right? Half a ton of the heat. That's how much energy we would need if we were as power hungry as a bacterium. Yeah. Oh, yeah. You just made my brain hurt. Okay, so if it's 1 kilogram, it's 10 to the minus 3 cubic meters. 10 to the minus 3 times a megawatt would be a kilowatt, which is 10 times our own metabolism. So the only conclusion is, right, the numbers that we call bacteria are bacteria that are living at really low density in a flask where they have an excess of growth medium and they can just make baby bacteria as much as they want. And when I had the bacteria and the biofilm, right, they had a much lower metabolic rate than the ones in isolated bacteria. So this is and actually the bacteria are probably a bit smaller than this. The bacteria, the 10 to the minus 8. The 10 to the 6 was around 10 to the minus 8, so micron. They're a little bit bigger, so they're not quite a megawatt, but still. So some combination of me overestimating and the density matters. Yes. Yeah. Yeah, exactly. Yeah, so I mean, I think that's a profound question, right? I mean, it's the question at the core of biology and active matter. And, you know, and the other thing too, I just want to emphasize again, like when I said on these plots, when they say bacteria, they mean bacteria under a certain set of conditions. So the thing that's crazy, I'm going to take a step back. Bacteria have probably metabolism, no, I'll say conclusively, a single bacterium can have whatever metabolic rate it wants. That's the amazing thing about bacteria. So if a bacterium growing under like really optimal conditions where that was measured will divide, you know, every few minutes. But when they don't have enough sugar, they can turn off their metabolism and live for 300 years. And then, or almost live, or sort of live for 300 years, and then turn their metabolism back on and grow when the conditions are favorable. So I think it's maybe the answer is for bacteria, because they're so opportunistic, and when they have a chance to divide, when the conditions are right, they just go crazy to divide. I think that might be one important factor. But I know from looking at these papers recently that even with human cells, that if you take an individual human cell, isolate it from a human or another mammal, and you grow it in a dish with excess nutrients, it will have a metabolic rate, that, if I remember correctly, that's significantly higher than the metabolic rate of it in the tissue. So I don't know why. Yeah, it's the data. Yeah. Sorry? Yeah, so people make claims about the metabolism in cancer cells, and I'm not an expert in this, but sort of just repeating what I hear people say. I guess that's what I almost always do, but in this case, I know less than I even normally do. But normally, people say cancer cells have a higher metabolism, and that's a lot of chemotherapies are designed based on trying to take advantage of the fact that they have a higher metabolic rate. But I told you more than I know already. Comments? More comments? So yeah, so this is a lot of fun data, right? I mean, and I don't think people understand this stuff. But for me, and let me just say, why do I care? Why am I going through all the trouble to calculate all this stuff? Because in my lab, I'm trying to create active materials that show some of the vitality of living active materials, and I'm like, well, I want a control parameter. Are my active materials active enough? And all living cells are between here and here, from 10 to the 3 watts per meter cubed and 10 to the 6 watts per meter cubed. And so I'm using that as an important control parameter in the active systems I'm designing in the lab to try to get in that range. And that's why I care about it so much. Okay, fun fact. I'm gonna give you the power densities for a lithium ion battery, sort of running, at optimally the way it normally, discharging happily, not short-circuiting, but being used. A lithium ion battery, and the average power heat generated by the sun per unit per unit volume. Does anybody want to venture an opinion about either one of those numbers? Okay, so I'll tell you that a lithium-ion battery, it has when it produces heat at about the same rate per unit volume as a bacterium, what I'm calling a bacterium, a megawatt per meter cubed. Okay? What about the sun? If you take the average, just like you do for a person or a bacterium on the allometric plot, how would you calculate it? I'll tell you, how would you calculate it? I calculated it. And I'm a soft matter physicist, so I didn't do any nuclear physics. What did I do? I used Stefan's Boltzmann. I Googled the temperature of the sun at the surface. I Googled the radius of the sun. I calculated the area, multiplied it by sigma t to the 4th, divided it by the volume, and I got a number. Anybody want to guess what number I got? So that's not the number I got, but that's the number on Wikipedia, actually. It's not the one I got, but the one I got, so yeah, it was 1 watt per meter cubed. What? That's crazy. And Wikipedia says 200 watts per meter cubed. And that's still, what? Right? I think the real, our metabolic rate is either with my calculation or, Wikipedia, which must be right, it has a much significantly lower metabolism than you do. So why, what's the obvious, so why is the sun so big and red and fiery? It's just so damn big. Yeah, and we'll get to that later. So it's pretty, imagine if you're producing that heat, throughout the whole volume, it's got to get to the outside, and it takes a finite amount of time for it to get outside. So Neil deGrasse Tyson in Cosmos said, which blew my mind. He said even that like a photon generated in the middle of the sun like takes like some outrageous number of years even a photon takes forever to get to the surface of the sun. So that's why it gets so darn hot. Okay. Anyway, but this number, it's like it's nuclear reactions and it's only a watt per meter cubed. Wow. It's really a slow burning object. Really, really slow burn. And that's good, right? We don't want the sun to increase its metabolic rate. Okay. Okay, so now what I want to do is, so we have a sense of scale in terms of my target activities for engineered systems, or you can think of them as living systems. Now what I want to do is I want to take these scales and try to come up with sort of various dimensionless measures of is this a big metabolic rate or a small metabolic rate. And now I'm just going to do a bunch of very simple back of the envelope style calculations which is the, as an experimentalist, most of the calculations that I do. And back of the envelope calculations are most important when you're trying to design an experiment. I don't bother writing down differential equations or boundary conditions or any of that stuff until I have data. And then after that, after I have data, that's when it's time to write down the differential equations. But at first you just do this sort of really simple stuff just to try to develop an intuition of the scales. And that's probably sort of the biggest difference in my talk from the theory talks is, for me, everything's about numbers. I always care about how big everything is. The numbers always matter as an experimentalist. All right. So the first thing I'm going to do is I'm going to try to come up with some picture of making some way to make sense of like how much is 10 to the 3 or 10 to the 6 watts per cubic meter. And I want to sort of think at a molecular level, I want to just sort of like, well, how many ATP hydrolyses is that, in some volume in a second, right? It's just a simple thing, okay? So an important fact is that ATP hydrolysis, anybody know how much energy it releases? Yeah, good. That's about right. You can see a range of numbers that people say. I like to use 20 kBT because it's easier to write than 25. But you'll see estimates from 20 to 30. And I mean, they're not estimates you can measure, but the point is that it really depends strongly on the concentration of phosphate and the concentration of everything else. But 20 is a reasonable estimate. And so if you just take that Q dot that I gave you and I divide it by the heat from ATP, I get another number that doesn't tell me anything. 10 to the 22 ATP hydrolyses per cubic meter per second at 10 to the 3 watts per meter cubed, which is like the human metabolic rate, and 10 to the 25 per meter cubed per second at 10 to the 6 watts per meter cubed. Okay? So I was trying to think, okay, this is just a big number. I want to get some physical intuition for what it looks like inside of a cell and what's happening. And so one thing I thought about is to ask the question is, what volume do I have to look at to see one ATP hydrolysis per second, right? Do I have to look at the whole cell to see an ATP hydrolysis in a second, or do I have to look at some much smaller length scale? And I got a volume of 40 nanometers cubed at 10 to the 3 watts per meter cubed, and a volume of 4 nanometers cubed at 10 to the 6 watts per meter cubed. Okay, so I know what a nanometer is. Okay, we all know what a nanometer is, but to really make to really bring this home, the key thing is that you need to know to interpret this, to build a picture inside of your head of what is happening inside of a cytoplasm, is that a typical protein, now proteins are, there's tiny proteins, there's huge proteins, but a typical globular protein is about 10 nanometers across. Okay? So now I think this is enough, this should be enough information for you to sort of imagine, the cytoplasm and all the proteins, make it easy, make them spheres, right? And you can imagine. And so here in a bacterium, in a volume a little bit smaller or comparable to a single protein, there's about 1 ATP hydrolysis per second, okay? And, or another way to think about it is there's, 10 kTs worth of energy getting released inside of that protein every second, right? So, there's a lot of energy getting dumped into that volume. Inside of a human cell, inside of your body, it's a significantly bigger volume, but it's just, it's just a factor of 10 in radius, so there's, a lot more proteins involved, but it's still a pretty small volume, okay? So I think that's a nice sort of intuitive way, one intuitive way to think about the metabolism molecularly. Cheers. He told me in advance. I told him that when he left, he should have said, that's crap, and walked out. But it would have been much more fun if he had done that instead of waved politely. Okay, any comments about that? Okay, please. Yeah, I don't know whether, we'll have to go look. I don't know if I have numbers for mitochondria or not. So that 10 to the 6 might actually be for mitochondria, not for bacteria. It was a log-log plot, and and I don't remember if the last data point was really a mitochondria or a bacteria. I'm not even sure if mitochondria are on that plot. I have all the references. We could talk about it later. Yeah. Yeah. Mm-hmm. But so you think this number is way too big? I'm trying to interpret your. Yeah. Yeah. It sounds slow to you. Yeah. Yeah, it does sound slow because if you look at I think the point is that if you look at the catalytic rate of enzymes, right? The fast enzymes can do hundreds of ATP per second, right? But then again, there are lots of proteins that don't have any enzymatic activity at all, and this is just the net balance. And then a lot of the mass of a bacterium is just in DNA, and the DNA is not doing anything unless it's replicating, right? So there's a lot of stuff, and it's not all enzymatically active, and there's slow and there's fast ones, slow ones. Yeah, 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 yeah. Well, no, this is just averaged across all of the proteins in the cell, some of which have zero metabolic activity, others, yeah. It goes way faster than 1 step per second. It's just an average. Are yeah, yeah, yeah. Think about it. We can talk more later. But actually, what's interesting is it actually, actually fits pretty well to if you. So you've seen these movies of like Kinesin walking, it takes about a step a second. So actually sort of kinesin would be sort of more of an average protein in terms of its metabolic activity than one of these spinny things on a bacterium that you're referring to. But other enzymes are, blazingly fast. So, yeah, I'd love, maybe it's in cell biology by the numbers, but it wouldn't be great to just see a histogram of sort of the metabolic rate of like, how many proteins have a metabolic rate of X, right? I don't know, I bet it's a power law distribution. It's bet it's got a long tail, but I'm just betting. Maybe it's minus 3/4. Yeah, I don't know. It's Friday night, so let's talk. Okay. Yeah. Well, both, I would argue. I mean, to first order, I'm doing it because I can do it. But look, anytime you're trying to understand, you start with, 0th order, first order, second order, and this is the mean of the distribution, and, yeah, it's the first step. I argue it means something. What it means is for you guys to figure out. That's your job. I'm here. I'm the experimentalist. I'm giving you numbers. You come back, tell me what they mean, okay? All right. Now let's talk about another measure, okay? I like this one. Let's think about it instead of a molecular basis. I want to think about it as mechanics, okay? So how much energy is there mechanically in this, okay? So in this way, I want to ask myself, how far, if you were taking all that energy and you put it directly into deformation, how far can you get from mechanical equilibrium? Right, so I'm assuming all the energy goes into mechanical deformation. And I'm going to consider two limits. One is where I treat the system as a viscous fluid, as a Newtonian fluid. And the other limit where I'm going to consider it as an elastic solid. Okay. Now, for a viscous fluid, well, for this case, we know exactly what Q dot is. What's Q dot for a viscous fluid? Yep, times. Exactly. It's like the only formula from fluid mechanics I can ever remember. Right? It's nice and easy, right? So when you shear a fluid or when the fluid flows, you're converting moment, and if it's a creeping flow, this is the rate that you're dumping the macroscopic flow into heat, right? So now if we imagine we're sort of doing this in the opposite and we're generating a shear from the energy that's released here. I'm going to show a table now. I set a plot. I'm going to calculate the shear rate. And here on this, I'm going to have two different values of the metabolism and two different values of the viscosity. So Q dot, again, is in watts per meter cubed. And we're going to look at always 10 to the 3 and 10 to the 6th. And then we're going to look at viscosity here. Viscosity, units Pascal seconds. We're going to consider two viscosities, the viscosity of water, because how can you not do the viscosity of water? And we're going to do the viscosity of glycerol, which is 1,000 times higher and a bit closer to the nominal viscosity of the cytoplasm. But of course, it doesn't make sense to really talk about the viscosity of the cytoplasm because it's a power law fluid. But when people try to measure viscosity and they ignore the fact that it's a power law fluid, they get something like that. Okay. So if I just use that, what shear rate do I get? Okay. So for our metabolic rate with the viscosity of water, you can get shear rates of 10 to the 3. That's a very, very, very high shear rate, right? So that's if I have a gap between this piece of paper and the table and I move this at a meter per second, that's a shear rate of 10 to the 3. It's a huge shear rate. And of course, if I do it with the bacterial one, I get it something 30 times bigger because the square root of 1,000 is 30. Okay, and even with the viscosity of glycerol, right? I can get a shear rate of 30 or a shear rate of 10 to the 3 um, from that. So it's clear like that this is this is an amount of energy that can really deform material, and for viscous fluids, I think this is pretty fast. Okay. All right, the elastic solid case, you've got to sort of turn your head to the side to make any sense of it, because elastic solids don't have an inherent time scale. And so what's the trick? So what we're going to do is we're going to say, okay, well, what's the energy density for a solid? Yeah, but in bulk, this is the energy density, not q dot, just q, little q. G gamma squared, right? So if you're trained as a classical, like as a physicist, you might have never seen continuum mechanics. If you only know remember two formulas about fluid and solid mechanics. These are two good ones to start with. Okay, yeah. So that's excuse me. Yeah. So so that's the energy stored in a solid. Okay. Now I imagine that to get there, I have my q dot and some amount of time that the thing has to work to achieve that strain. And if I just arbitrarily let's just set gamma equals 1, right? So then what we're really going to calculate then is the time it would take at that metabolic rate to achieve, this should be G prime, not G, G prime divided by Q dot, okay? That's sort of a nice intuitive way of converting it into, thinking about elastic solid. So how long does it take to get a significant deformation, okay? So same type of plot table as before. And we have Q dot here, which is 10 to the 3, 10 to the 6. Same, I won't label it again. And then G prime here in Pascal. And we're going to use 10 to the 3, which is sort of a typical cell modulus. Again, you shouldn't really give it a modulus because it's a power off fluid, but if you pretend it's a solid, you typically measure something like a kilopascal for a typical cell, okay? But muscle cells or muscle tissue are about 10 times higher. So if you take sort of muscle tissue or a muscle cell, you get about 10 kilopascal for a modulus. And then if you say, okay, if a cell with a modulus of 10 to the 3 has this metabolic rate, then it takes about a second for it to develop a strain of 1, right? So, that's pretty awesome. And if it was a bacterium, it could do the same thing in a millisecond because it's 1,000 times faster. And then, but for muscle though, muscle has a stiffness of about 10 to the 4th. It takes about 10 seconds to at that metabolic rate. Oh, sorry, this is tau in seconds. This table is writing down tau of seconds. But if it had the bacterial metabolic rate, it could do it in 10 milliseconds, right? So this is another point. This is related to this thing you were saying. So you said, but the thing's spinning around really fast. If the muscle had the average metabolic rate of my whole body, then to move my arm like this would be 10 seconds. Right? So I would say when I go from here to here, this muscle is having a strain of order 1, right? If my muscle just had the same exact metabolic rate, they had the average, I would move like this, right? All the time. But the metabolic rate varies tremendously from different parts of our body. The same thing I would say is happening inside the cell. Comments? Okay, so these are a couple of ways. One is sort of molecular, one is continuum of trying to make sense of these metabolic densities that I gave you. Now I want to shift gears a little bit and sort of ask some philosophical questions. They're not philosophical questions. They're fundamental physics questions, but they're so far from being answered that they feel like philosophy. But hopefully one of you will be able to tell me all the answers. If not today, then soon. Okay? So I'm going to call these motivating questions. Okay. So this is for me the sort of the driving physics question of all of this stuff. Okay? So how much can we say about an active system when we know only Q dot? Right? Like, can we put any constraints at all, or can we say anything interesting about a system only knowing this flux, okay? And, or maybe a little bit better is what if we only knew Q dot above plus the fluxes, the mass fluxes of all the chemical components. Right? So what I'm really asking, right, is I'm saying I'm accepting that I don't know the whole that I don't know a lot about my system. I'm accepting the fact, I'm accepting my ignorance. And are there, even though there's so much I don't know about the system in these two cases, is there anything intelligent we can say about it? It sounds like it might be a crazy thing to do, but right, we have thermodynamics, right? In thermodynamics, we know almost nothing about these systems that we describe using thermodynamics, but we have very general and powerful rules for putting constraints on what's possible. And so I guess my real broad question, right, is sort of stated and sort of slightly differently, is like, are there general principles akin or similar to or inspired by, maybe they won't look anything like thermodynamics. Maybe, there's no point in calculating an effective temperature most of the time. But, just in the spirit of thermodynamics, where we try to say the most about a system while knowing the least, that put constraints on active systems or materials. Okay. It seems really aggressive for me to even suggest that anybody might be able to answer this question, especially in the last, since what we've been hearing over the last few days from me and from other people. I showed that, when you were changing the activity of the cell, it really made a difference if you were just getting rid of myosin, or depending on which protein you get rid of. So the molecular details mattered, at least in those particular cases. And then Ludovic yesterday, argued that, yeah, you can calculate an effective temperature, and sometimes it does something for you, and other times it does absolutely nothing. And but maybe we just haven't done it the right way yet. And the thing is, if I just change this question, it says, are there general principles akin to thermodynamics that put constraints on active systems? The answer is yes. We call it thermodynamics. In fact, that was the whole reason thermodynamics was invented in the first place, right? Because people wanted to figure out how good a heat engine could be, right? And so even though heat engines operate really, really far from equilibrium, they still put useful constraints on how things operate. So is thermodynamics enough? Probably not. I think you need more to understand, because I don't want to just put a limit on the efficiency of a living system. Maybe that I should try to do that first. But I would also really be able to like to have the equivalent of a principle saying, if I want to know the structure and properties of the material in equilibrium, I would minimize a free energy. Is there something like a free energy that I would do something to, like minimize or something else, that would allow me, given my metabolic rate and other generic features, to predict the structure? And I think that's really what we're ultimately all here for, and we should not forget that, right? And let's not get too stuck in the details of our favorite experimental system or theoretical system. Let's keep the big questions in mind, okay? And this gets back to what is life? This this amazingly prescient little book by Schrödinger, where he predicted all sorts of things. And I want to read something to you from Schrödinger. I'm not going to write it on the board; it's too long. But you can Google it. It says it emerges that living matter. What? Whoops. Sorry. Oh yeah, sorry. Yeah, it emerges that living matter, while not breaking the laws of physics as established to date, is likely to involve other laws of physics, hitherto unknown, which, however, once they have been revealed, will form just an integral part of this science as the former. Okay, so Schrödinger in 1944 really argued that to figure out how cells work, we need new laws of physics. Okay, so find them, please. They're there. Okay, and you should, as a young scientist, you should aspire to finding new laws of physics, not just new phenomena. Right? Go for it and be general and change the world. Okay. Okay. Comments? What did he mean? So he was, I don't know. It was right, it was sort of it was the last paragraph of the chapter on entropy, and how the idea where he promotes the fact that living systems are just systems that produce entropy, and that the reason we don't eat for the energy, we eat for the entropy is the argument that he made. Which he has a footnote saying, well, maybe I should have said we eat for the free energy instead of for the entropy, but whatever. He was making an argument. But anyway, so it was after talking about all this stuff about the relationships. It was right after he talked about the nature of thermodynamics and life that he made this statement. So I sort of feel like he was really talking actually about this, actually. That's my guess. I don't know. I'll read it again. I pulled the quote out of context, so I don't quite perfectly remember the context. Okay. So that was supposed to be the last thing I said for like my lecture, my lecture just before the break. Okay. So this might be a good time if you need to take a 5-minute break to go to. Yeah, let's just say 5-minute break so we can all ponder. What Schrödinger said, and go to the bathroom, and then we'll move on to the next thing. Okay? little bit for my last sort of like, inspirational speech or attempt at being inspirational speech about the big questions in physics and biology, and still motivated by that. I want to be able to do experiments that help you figure out those theories that and get all the credit for 100 years from now, because the theorists always get the credit. Anyway, that's okay. I don't mind. Anyway, so now we're going to start talking a little bit more about sort of experimental design and the things that I'm worrying about as I try to build the experiments that actually can get good enough data that you can make theories that work. Okay, so I'm going to call this next part toward active materials. And again, I said the thing that distinguishes active materials from active systems is that they're steady state, they're homogeneous, and they're in space and time. So I can start going about defining properties and measuring properties as a function of various metabolic parameters. And the key thing, which I've said several times, but it's always good to say things multiple times, is that steady state requires fluxes. And if we think of our active material or our active system as a black box, there's some flow of mass going into it, which we call the fuel, and there's a flow of mass coming out of it called the waste. And in the process, it's going to reject heat and to the surroundings and potentially do work on the surroundings. Now, if you really want to build an active material, you have to spend some time thinking about these fluxes. Okay, this microphone, I don't know how to get it properly around my ear, but anyway. Yeah, you have to really worry about the fluxes, okay? And the key point here, and I'll just say the point now, is that the transport of material doesn't happen, at the speed of light, right? And usually doesn't even happen with the speed. It happens diffusively. And so the point is that if you want the system, because the material's going in and out, the heat's going in and out, that transport actually is going to limit the size of a system if you demand that it's homogeneous. Okay, remember, living systems are not homogeneous, but I want to make experiments as materials in my lab that have the same metabolic rate as living systems but are homogeneous. Okay, and to really have it be a material, what I need, my criterion, is to be a material. The maximum size limited from the transport phenomena, from transport, needs to be much bigger than the structural scale. Okay, so anytime you have a material, there's, if you're above the structural scale where you see the molecules or atoms or colloidal particles that make it up, if you zoom out far enough, that it has extensive properties that are proportional to the volume, or it has intensive properties, right? So that you can treat it as a continuum is basically all I'm saying. Is there really a continuum limit for active materials? In order to achieve that, the maximum size due to transport has to be much bigger than the continuum, the structural scale. Does that make sense? Okay. Gerhard, can you take a look at this? Because I can't figure out what I'm doing wrong. Okay, we'll see. If not, then you feel free to, because I've done everything I can just by grabbing randomly. It looks good? Okay, all right. Super. Okay. Yeah, okay. So what I want to do is I want to consider various transport phenomena associated with these mass fluxes and the heat flux, and ask, are they big enough for B to be able to make a material? Okay. So I'm going to propose a a minimal active system. Okay, a minimal active system. Minimal active system. Okay, it's going to be a system where the activity is driven by the consumption of fuel. Okay, and I'm really bad at chemistry, so I'm going to call the fuel F. Okay, F for fuel. And imagine this thing works is the fuel interacts with some catalyst. You'll see in a second why I need the catalyst. Interacts with some catalyst to produce waste, which is some other molecule. And the catalyst is unchanged. Okay, that's what you mean by a catalyst. This reaction only takes place in the presence of the catalyst. And yeah, so in a living system, right, this might be sugar, right? And then this would have to be more than one thing. Not just one thing, okay? But we're just keeping it simple, okay? Okay, so what we want to look at is we imagine that our active system is a sphere, okay? It has some radius r, and the catalyst concentration is greater. So catalyst, so phi c is the catalyst. Well, I'll just say phi I are the concentrations, right? Phi c is the catalyst concentration. Inside of this sphere, there is catalyst, okay? And outside the sphere, there is no catalyst. So all the reactions that take place are going to be localized to the sphere, okay? And then infinitely, some distance far away, at little r goes to infinity, okay? The little r is measured from the center of the sphere. There is, we specify some concentration of fuel at infinity. We'll call it phi f infinity. We specify for now that the concentration of waste is 0, infinitely far away, and that we have some temperature T infinity, okay? And we're going to assume, as you can see here, you can infer here that there's no flux of C. The catalyst has to stay inside of the sphere, but at the same time there's free diffusion of fuel and waste at the interface. So we don't have to, we won't worry about membranes or anything like that, and they're unfashionable anyway, okay? So that's the basic setup, and the basic question we want to ask is what are the steady state profiles of the chemical components and the temperature. And in what region of parameter space do I need to be in order for this thing to have a homogeneous composition and temperature? And the answer is that it's going to have to be pretty small, okay? And we're going to see, but the question is, how small does it really have to be? So I'm going to erase this. Try to think of some questions about my setup. If you need any questions about the setup, yeah. Yeah, Exactly. There's no reason except that it's harder, right? It's just more stuff. I want to self-assemble my active material. I don't want to go to a clean room to make my active material. In fact, you know, if I have to go to a clean room to make it, it's not a material by my, well, then it's people would call it a metamaterial, right? But I want to make a real material, right? So I don't want to have to put stuff in it. But that's it. I mean, this is, I was going to make this point later. You know, I'm going to find some. I'm going to assume diffusive transport everything. I'm going to get an answer that's pretty small, but but big enough to be workable, which is good news. But you know, like we have circulatory systems, right? So the networks of tubes are good things. Yeah. Yeah, I'll explain it with words. I didn't really explain it too much because the calculation I'm about to do is going to really explain it and like really explain it, but I'll say it in words. The basic idea here is that this thing is eating up the fuel, right, inside the sphere. You drop it into a bath of fuel, and you can imagine if the reaction rate between the fuel and catalyst is super high, or the catalyst concentration is super high, that the fuel might not diffuse very far before it gets converted into waste. So we want to find ourselves in a limit where the fuel can diffuse all the way across it, or at least to the middle, before having a significant probability of having of being consumed. In the extreme limit, it would all the fuel would just get consumed at the interface, right? If the reactions were really, really super fast. And then we'd have an active interface. We wouldn't have an active material. That'd be a cool experiment, but it's not what I want to do. It's closer to like a Janus particle. Yeah. Yeah, yeah. Yeah, so that's, you know, like that, you know, that's been a subject of, you know, that was like one of the central questions of 20th century chemistry, and it's still an active question. How do catalysts work? And the, you know, the very my naive physicist understanding of how catalysts work is that, you know, you have a reaction that wants to proceed forward. So you have a free energy landscape, right? So this is fuel, this is waste. There is a significant delta G between these two, so this should be a spontaneous reaction, but the energy barrier here is big. And so the idea is when the fuel, somehow when it sits next to the right catalyst, that lowers the barrier and that it increases the rate of the reaction. So that's the basic idea of what people... There is, and we'll talk about in a second, but the simple, there's a whole ton of physics in our chemistry there. There is one intermediate step that I'm about to tell you about, is that you just need to have binding. At the first level, the intermediate step between these two would be FC. I'll write that in a second. But yeah, the two have to bind at a bare minimum. I am going to put in one intermediate step, the binding, and that's all I'm going to do. Yeah, but and you know it's different. But the thing about catalysis is it's like there's a trillion different ways of achieving catalysis. There's no one way of doing catalysis, but minimally there's got to be some interaction. So there is at least one intermediate, and basically it's reducing a free energy barrier. Okay. Okay. Great. So let's talk about catalysis now. So rates of catalyzed reactions. Okay, so now I'm going to, you know, so this is the degradation of F with the help of C. And the chemical formula is exactly what you're asking me for. There's F plus C, generally thought to be an equilibrium. As there's reversible binding between the fuel and the catalyst. But once the fuel has been bound to the catalyst, it produces the waste, and the catalyst is liberated, and it's free to bind to someone else. Okay, so this is sort of the classic Well, soon it's going to be super classic, but this is one of the simple ways of thinking about catalysis. And there's a rate, Kf. This is sort of the rate of how many moles per cubic meter per second do you make of Fc. And there's a forward rate and there's a reverse rate here. And there's also a rate that you produce waste once you've already made the complex, and that's called kcat. I think it stands for catalyst, probably. Okay. So the question is, what's the overall rate of the reaction, given that we have these intermediate rates? So the overall rate, I'm going to call, I actually was scratching my head. I couldn't figure out why I call it, but I'm going to call it S. And it's going to be equal to the rate that we increase the concentration of waste, which is equal here in my simplistic stoichiometry to the rate at minus the rate at which we're consuming the fuel. Okay? And in certain limits, it's actually really easy to derive if you make a couple of assumptions. So if you, and I won't go through the derivation, but the derivation is on Wikipedia. And once I tell you what this is called, well, I'll tell you now. Michaelis and Menton. If you make the assumptions that all you have to do is you assume that in addition to this reaction scheme, you assume that the concentration of the fuel is much higher than the concentration of the catalyst, then you get this equation for the reaction rate. It's kcat times the concentration of the catalyst. So this part is all just about the catalyst, property of the catalyst. And this other part is phi f over km plus phi f. Sorry, f is the fuel, where km depends on the binding affinity of the fuel for the catalyst. Okay. And I'll just give you really quick the before I plot the answer. Km is equal to Kr over Kf, so that high affinity means high affinity between the fuel and the catalyst means a small Km. All right. Yes. Oh, it's very simple. This arrow goes both ways. This arrow only goes one way. That's a really pithy answer to this. So the idea, right, is that this is a reversible process. Reversible processes can reach equilibrium. Irreversible processes produce entropy. So once you get here, boom. And I was just about to, when you go here, you release a ton of free energy and doesn't go back and create heat, create entropy, blah, blah, blah. So that's why it's active. No, it comes from the fuel. So like, somebody really far away gives you the fuel, and the catalyst is coming around and is lowering the free energy of the fuel. That's the way I think of it. There's a ton of free energy in the fuel. Catalyst is lowering the free energy of the fuel. Lowering the free energy of the fuel allows it to do work. I think I saw a question over here. And what? Why they're different? Yeah, so it all depends on the binding affinity between Kr and Kf. So this is actually, the Km is actually the equilibrium constant for the binding of these two, and so it is, it's proportional to e to the delta g or minus delta g, depending on which way you define it for the binding of the two. So it's definitely related to the energy associated with that binding. Yeah. Uh-huh. Yeah, I mean, I guess that's right. Because so if I just sort of, if I sort of seal, if suddenly I let the fuel in here and I sealed off the container, eventually the system would reach equilibrium, and that might correspond to some reverse current. You're right, but by the fact that the waste can go away and more fuel can come in, it never gets a chance to establish equilibrium. Yeah, thank you. Good, good comment. More people should make comments like that. Why do I have to do all the work? It's my Friday night too. Okay. So yeah, okay. So what does the answer look like? So I gave you the answer, but it's always better to draw a picture. Well, not always better, but it's highly complementary to draw a picture. So if we plot the reaction rate here, like this, and on the horizontal axis we plot the log of the concentration of the fuel, then you get what the biologists would call, or a sigmoidal curve. I don't know why I decided to make it fat. Maybe just because it's almost the end of the lecture and I'm getting tired. Anyway, so that's what the reaction rate looks like. That the key point is below some critical concentration, where phi m is equal to the equilibrium constant. That if the fuel is lower than this, right, the fuel concentration is lower than this, there's basically no production of waste. And the way to think about that is there's just not enough binding between the fuel and the catalyst if you don't have enough fuel. But if you get above Km, then you get a lot of binding of the fuel to the catalyst, and just the reaction runs forward. Okay, so there's a critical concentration. And if you're significantly above Km, the rate is constant, and the rate is equal, I'll write the equation in a second, but it's equal to kcat times phi c. It saturates. Eventually, the catalyst is getting a binding to fuel. There's so much fuel, as soon as it produces waste, it can bind another fuel molecule. Okay, so there's some limiting cases that are important. There's the one I just said, right? So when phi f is much bigger than Km, with the fuel concentration is much bigger than the critical concentration, S saturates. It's kcat phi c, and I call this limit, I call this catalyst limited. It might be what other people call it too, but I didn't read the literature well enough to know if that's what other people call it. And if phi f is much less than Km, it's not strictly 0, it's just small. And then S is kcat phi c, phi f over phi m. It's a little bit complicated, but the key point here is that here the reaction rate is independent of the fuel concentration. Here the reaction rate is proportional to the fuel concentration, okay? So I call this the fuel limited case. You could call it the fuel limited, or you could say it's diffusion limited. I think sometimes that's maybe a more conventional thing to call it, the fuel limited case. Now, this form, this simple expression, like picture of catalysis, is the one that is mostly used by the biochemical community. And so if you've heard Michaelis-Menten kinetics, you probably heard it from a biochemist. And as I mentioned before, it's really important to be able to speak biology, right, and to translate back and forth. So, so in a biological context, what do people call the catalyst? Enzyme, yeah, great. And then what do people call the fuel? The substrate, which confuses the crap out of me. Because for me, a substrate is something you put something on, but it's fine, it's okay. It's okay. And well, they get completely confused by my substrate, so it's okay. Equally confused. And then the waste, they have a more positive way of describing it. They call it the product. Okay. Oh, that's the empty one. Good. The product. Okay, so if and then instead of S, they would say V, but they would normalize it differently. Their V is also, I can't remember what's different about how they do the V. I think they do it per enzyme. I think they calculate the rate per enzyme. Yeah, yeah. So S would be V times R, the phi time, the concentration of the enzyme. Yes. Okay. Good. So anyway, that's the Google Translate. Okay, between physics and biology. Questions about that? No. Okay. 15 minutes. I'm looking for a good break spot. I think this is a good break spot. It's Friday night. Would anybody really get mad at me if we end 15 minutes early? No? Okay. Okay, the lecture is now unofficially over. I will stop talking in front of everyone. I will turn off the thing, and I will be happy to discuss anything from now through dinner. And the lecture will continue during dinner, okay? And at the bar later. Okay.